Swayam Prabha. Digital India. Educated India.
Welcome to my course Industrial Biotechnology. In the last class I discussed the air sterilization, and I told you that biochemical industry that the sterility is a very important factor, because we shall have to allow the growth of a desired organism in the reactor, so we should not allow any foreign organism to enter into the process. And for that we find two important the source of contamination. One is air, because most of the fermentation is carried out under aerobic conditions, and media that is required for the growth of the microorganism. So in the last class I tried to share that air sterilization, and main objective was to remove the contaminants that present in the air. Now when we design any kind of air sterilization process, that we always assume that a basis is that out of how much of contaminants, how much organism you are going to remove. As for example, whether you are removing 1 in 1000, 1 in 10,000, 1 in 100,000, like this. So you have to, the more your process is stringent, your sterility is requirement is more, you take more precautionary measure. And another issue I told you that the type of organism you remove, again it depends on the type of fermentation you are carrying out. I have given the example of citric acid fermentation process. We use the cane molasses as a raw material for citric acid production. Now cane molasses also a very good raw materials for yeast fermentation process. Now if you look at the doubling time of yeast is much less as compared to doubling time of that Aspergillus niger, which produces citric acid. So naturally contamination of yeast is much more concerned as per the citric acid fermentation. So and usually in the air filter we use the physical separation technique. We use some kind of depth filter to remove the contaminants. Now it has been chosen that a glass wool fiber is considered the best for the removal of the contaminants present in the air. The reason is that here drag coefficient is quite low, and not only that, you can regenerate the glass wool fiber again and again, because when after some time when you use glass wool fiber as a filter, after some time your filter bed will be totally saturated with the contaminants. So in that case what you have to do, you have to remove the contaminants. Now question comes that the size of the air filter again depends on the quantity of air that you are going to sterilize. Suppose in case I told you that the size of the inoculum vessel is usually 5 to 10% of the production fermenter. So naturally when we prepare the media, we design the air filter for the inoculum vessel. Our size of the filter requirement will be very less, but when you go for production fermenter, it will be very high, because size will be 10 times higher than that of the inoculum vessel. So now question comes that I can show you the very simple design of that air filter, that is we have in practice in the industry, that is like this. We have this is the air filter we can have, and here we should have some kind of stainless steel mesh just to protect the fiber. Now you pass your air like this, and this is air in and this is air out. This is how we can do that. Now, this is for inoculum vessel, it is fine because this kind of small system we can use. Now, when you go for the production fermenter, so the size will be 10 times higher than that because the capacity of the production fermenter is more. Now, question comes that if you increase the surface area like this 10 times, it is very difficult, it will occupy a lot of space and it is very difficult to design. Be difficult to control the process. So what we have to do, we shall have to make the process air filter very compact. So question comes how we can make this air filter compact. Now if we have some cubical things that like this air filter, so we can have this like this inside. So if you pass air inside and air can comes out like this in different direction, you can from the annular space you can keep hold the assembly inside the filter and you can increase the surface area. So so like this you can take you can take the air out from here. The annular space we can make the system very compact in case you require the higher surface area. So this I forgot to mention in the last class. Now today I want to discuss about the medium sterilization, and in medium sterilization the purpose is same, that we shall have to remove the contaminants that present in the water in the media, and this is the destruction or removal of viable organism with an object or from a particular environment. Now various method of air sterilization is there. One is the heat is the best media for media sterilization, because when we talk about media is the liquid media and heat is transferred by three different mechanisms. One is conduction, and this is convection and radiation. So this is all three processes will be very active in case of medium sterilization, but we have two methods. We have dry heat, we have moist heat. We have seen moist heat is much effective as compared to dry heat. I shall show you later. Then radiation method, then filtration method. Chemical methods we use for like alcohol, oxidizing agent, salt, surface active agent, and this is a chemical method is rarely used for medium sterilization. Mostly we use either heat or the filtration method. Now again, when we use the filtration method, I told you that heat, as you know, heat your media comprises of lot of nutritional component like vitamins and amino acids. So these vitamins and amino acids, they are very sensitive to temperature. So as you increase the temperature, the quality of this media will be lost. So in some fermentation process we required we shall have to maintain the quality of the that media. So in that case we shall have to the vitamin and other things we can filter through the filter paper just to sterilization purpose. And as I told you, when you filter, we use a membrane, and membrane has some pore size. And if you suppose I want to remove bacteria, the smallest and size of bacteria varies from 0.5 to 2 microns. So 2 microns. So suppose now if I keep the pore size less than 0.5 micron, then we can when you pass your liquid, then all the bacteria will be retained here and comes. Now only the problem with that, I told you that there will be tremendous pressure drop across the membrane, and the life of the membrane also quite less. So it is very expensive as compared to heat. Now when we consider heat as a sterilizing media, the question comes how heat is effective for sterilizing the media, because we know that organism when you heat the organism, that it contains a lot of protein molecule, and protein at high temperature is undergo the denaturation. As the denaturation of the enzymes take place inside the organism, that the biochemical activity of the organism will be stopped and your organism will be killed. Now we have thermal heat method, we have dry heat. It employs the higher temperature in the range of 160 to 180 degrees centigrade and required exposure to up to 2 hours depending upon the temperature employed. So this is very if you compare with the moisture heat, moist heat is involved the use of steam in the range of 121 to 140 degrees centigrade. Steam under pressure is used to generate high temperature needed for the sterilization. This moist heat is much effective than dry heat. That is why dry heat we require a lot of high temperature as compared to moist heat. Now sterilization by radiation can be achieved by the electromagnetic radiation such as the electron beam, X-ray, gamma rays or E radiation of subatomic particles. Electromagnetic or particulate radiation can be energetic enough to ionize the atom or molecule or less energetic. So, but non-ionic radiation like UV rays radiation is useful to sterilize the surface, some transparent object. UV irradiation is routinely used to sterilize the interior of the biological safety cabinets, because that is, we know in the operation theater also we use the UV rays for surface sterilization purpose, because in the biochemical industry also we have we have lamellar flow where we prepared our culture, that inside the chamber we usually sterilize with the help of UV rays. Now the radiation method is the ionizing radiation sterilization. Gamma radiation is very penetrating and is commonly used for sterilization of disposable medical equipment such as syringe, needles, food. It is emitted by the radioisotopes. Now second is the filtration methods. Filtration process does not destroy but remove the microorganism. That is why I told you that in case of heat sensitive material we use that, but this is very costly process. And when you talk about the biochemical industry, we have two type of products. One is called high value products and another is low value products. High value products means per unit cost of the product is very high. Here, because since the per unit cost of the product is very high, there we can think for this kind of technique where high cost involvement is there. But in case of low value product, we cannot, because low value and high volume products, we cannot think for this process will be no good. So it is used both for the clarification and sterilization of liquid and gases, as it is capable of preventing the passage of both viable and non-viable particles. Sterilized solution that may be damaged or denatured by high temperatures and the chemical agents. The major mechanisms of filtration are sieving, adsorption, trapping within the matrix of the filter material. The pore size of the filtering for filtering bacteria, yeast and fungi is in the range of 0.22 to 0.45 microns. So this is the size. The more we go to the lower size of the pore, the pressure drop across the membrane will be high. Now in case of heat sterilization, which is in practice in the industry, we have thermal death time. It is the shortest time required to kill all the microorganisms in a sample at a specific temperature and not definite condition. Now here I want to point out that whenever we design any kind of sterilization process, medium sterilization process for the industry, we never go for the 100% sterilization. We always assume 1 out of 1 million, 1 out of 10 million, 1 out of 10,000 like this, because as per the whatever as per requirement of the process. Now decimal reduction time, that is also is a kind of term we related to the medium sterilization, is the time required to 90 to kill 90% of the microorganisms in a sample as a specific temperature. So the time required to kill the 90%, or that means if 10 organisms is there, it will be becoming one organism. So this is the kinetics of thermal death of the microorganism can be expressed like this: dn minus dn by dt equal to kd into n, where kd is the thermal death rate constant and n is the number of viable organism presence. Now this is like this. I shall show you this. You see that if you look the in ln n this is the plot of ln nt. Nt means to that as the number of viable organism present at time t and initial number of organism, that is keep on decreasing. It is a sharp plot with respect to time. This is the that means is follow the first order kinetics. So this is like this. So it is like this. So we can write minus dn by dt equal to kd into n. Now what we can do that so we can write minus dn by n equal to kd into dt. Now this is equal to minus dn sorry, this is d ln n minus equal to kd into dt. Now if you plot, if you integrate n0 to n whatever you have, or nt whatever you have, this is 0 to t, then what will be this? Minus ln nt by n0. Am I right? This is equal to k into t. Now if you take this minus this way, this will be minus kt. In that case nt I can write n0 into e to the power minus kt. So the number of organisms present at any time t we can easily, and this is k is nothing but kd. So this is the kd. Kd is the thermal death rate constant. The unit of this, since it is follow the first order kinetics, so it is the unit is time inverse. This kd unit is time inverse. Now this kd we can determine experimentally, because there is some technique, because you have to take in small capillary. If you have hot and cold bath and you take the culture initially, if you take the instantaneously, if you cool and hot this material, then what will happen, you can find out that that how much viable cells is present by exposing a particular temperature for higher for a particular time. So you can easily calculate the kd value. Now how to calculate the decimal reduction time? Decimal reduction time means 90% removal of the initial microorganisms. So n by n0 will be 1 by 10, and if you write it, so we will be coming this d equal to 2.303 by kd. That means your decimal reduction time is inversely proportional with thermal death rate constant. This is kd, inversely proportional. So as the time increases, the kd value will increase, because I can show you here that here you can see this is like this. As the time increases, that this will be becoming more steeper, and like this. Now if you plot this log graph paper, then we have this kind of plot. This is in case of 60 degrees centigrade and 54 degrees centigrade. If more increase the temperature, it will be more steeper, so kd value will increases. So what I want to mean that as you increase the temperature, kd value will increase to a great extent, and your D value will decrease. Now, so this kinetic description makes two prediction which contradict with each other at definite time is required to achieve the sterile condition, and after certain time there will be less than one viable cells remaining. So, you know that this is thus a value of NT less than one microorganism remaining is considered in terms of probability of an organism surviving a treatment. So, I told you that that when you design any kind of sterilization process, always we assume 1 in 1000 or 1 in 10,000, 1 in 100,000, what sterility factor you are going to have in your system. Now, here there is an interesting table that shows the how temperature affect the value of Kd. This is the Kd value. This Kd is thermal death rate constant. As you know that as you increase the temperature 100, 110, 120, 130, 140 and 150, the Kd value drastically changes. This unit is minute inverse. The holding time means this is the time required for sterilization. I shall explain the what is called when I shall discuss the continuous sterilization process. Now, as you see, when you have we have the 100 degree centigrade, we may hit the media 100 degree centigrade, then Kd value is 0.02. You have to sterilization time is 1730 minutes. But 110 degree centigrade, the Kd value is 0.21, then it's reduced to 164 minutes. 120 degree centigrade, Kd value is for 2, that the sterilization time is reduced to 17 minutes. Now, 130 degree centigrade is 17.5, and this is the 2 minutes is the sterilization time. 140 degree centigrade is the 136 is the Kd value, and holding time is 0.25. Now, in the industry, that you know that in the laboratory we use our sterilization temperature is 121 degree centigrade for 15 minutes. This is the sterilization temperature and time. This is the time requirement is this. So, now in the industry that we use actually 140 degree centigrade, because now and usually that you see that your sterilization time is reduced to 0.25 minutes. The holding time is 0.25 minutes. Now, question comes why we go for this high temperature sterilization. So, in the industry we usually follow the techniques what you call high temperature short time techniques. Now, why we follow the high temperature short time techniques? The reason is that if you look at the EA value means activation energy value of the microorganisms are 250 to 290 kilojoules per mole, and the activation energy for vitamins and amino acid is typically 84 to 92 kilojoules per mole. That means if you heat the media at a low temperature, it is more effective for destroy the vitamins and amino acids. That is why you know that in the household purpose, when we purchase some kind of medicinal tonic, it is always prescribed that you should keep it at low temperature, so that those tonic mostly comprises of some vitamins and amino acids, and they are very sensitive to temperature. So even at low temperature, the denaturation may take place. But if you look at the higher temperature, that you have EA value is we can have higher that activation energy requirement high. So this means the small increase in temperature has a relatively greater effect on cell death than the nutritional destruction. This fact becomes the basis for the use of high temperature short time techniques in the industry. This is why that industry we follow the high temperature short time technique for the sterilization of the media. Now, we have two type of sterilization in the industry, that is we have batch sterilization. Batch sterilizing the entire volume of the media at once using the heating and holding the cooling methods. So you can have this is the media, and you can pass your stream like this, or you can electrically heat it, or you can have jacketed, whatever you can. So you can do the sterilization like that. This is called the batch sterilization. And continuous sterilization means sterilizing only a fraction of the volume at a time by using the temperature as the internal heat exchanger. Now, basic difference between the batch sterilization and continuous sterilization: in case of batch sterilization, the entire volume of the media at once, the heating, holding and cooling. So what does it mean? What does it mean? Suppose this is the fermenter, and you have media. Now you want to sterilize this media. So this is the starter we have, and we want to sterilize the media. So first you have to heat it. So you require some time to, suppose this is our ambient temperature is about 35 degrees centigrade. So you have to increase the temperature, and then it attains 121 degrees centigrade. Then you have to hold it for 15 minutes, that is like this. Then you have to cool it. Am I right? This is cooled to the again 35 degree or 40 degrees centigrade, whatever you have. So this is called heating. Then this is holding. You are holding the temperature, and this is called cooling. Now we have seen that different organism has different depends the death of the organism depends on the temperature. We have seen just now that as you increase the temperature, Kd value changes drastically. So we know that means at different temperature you have different Kd value. So there will be death of the organism during heating. There is the death of the organism during holding the temperature. There is the death of organism during the cooling. So you know that what the total sterility is a combination of all the three things, both heating, holding and cooling. That is the total effect we have. That is why if you look at that the basic difference between the continuous sterilization and the batch sterilization. In the continuous, medium sterilization is based on the concept of high temperature short time. This takes the advantage of the fact that an increase in temperature has a relatively greater effect on the thermal destruction of the cells than on nutrients. The steam consumption of continuous sterilization is perhaps 20 to 25% of the requirement for a batch cycle. So this is a very crucial factor, because whatever steam we required for sterilizing the media in a batch system, that is very high as compared to that of continuous sterilization process. The total time required to sterilize the temperature in case of continuous sterilization is 2 to 3 hours compared to 5 to 6 hours in case of batch sterilization process. So time is a very crucial factor, as you know, in the industry. So this is very effective, that the continuous sterilization process is very effective. One point I forgot to mention, that when I showed you the effect of temperature on the killing of microorganism, even if you increase the temperature from 140 to 150 degrees centigrade, your sterilization time again further reduce. Now question may be asked, why you are not going for very high temperature? Now you might be aware, in the industry we increase the temperature by increasing the steam pressure. Now if we have very high temperature, the equipment design is a problem, because some kind of stringent we have with respect to equipment design. That is why we in the industry, if we feel 140 degrees centigrade is most suitable for the sterilization of the media. So I want to stop here now, and next class I am going to discuss about the continuous sterilization process. Thank you.